Using GeneStix, a tutorial example. Brought to you by Princeton Separations.
GeneStix are the key element in Princeton Separations' new DNA purification method for extracting a DNA band, or bands of interest, after Agarose Gel electrophoresis separation. The following animation provides an example of how GeneStix may be used to extract DNA bands from gels. First, we'll prepare the agarose gel. Dilute the binding buffer concentrate in a volumetric flask. Weigh agarose to make a 0.8% gel and place in an Erlenmeyer flask. Measure out the binding buffer, mix with the agarose and microwave to dissolve. Pour agarose gel. Next, we'll load the gel with samples and markers. Samples are prepared with tracking dye. Samples contain 20 kb DNA with a 1 kb DNA ladder, and 20 kb DNA with 5 kb DNA. Cyber gold is used to create visible migration markers. Selected sample standards are premixed with cyber gold. Samples are loaded onto gel. Now, we'll see how to calibrate the migration gauge. Perform electrophoresis in the running buffer. Remove gel from electrophoresis device. Place gel under blue light illuminator. Align migration gauge with gel well. Using lane 1 standards, mark migration gauge at position greater than 3 kb DNA. Using lane 1 standards, mark migration gauge at position greater than 6 kb DNA. After calibration of the migration gauge, move the gel to room light. Move migration gauge to allow insertion of locator tabs in lane 2. Insert locator tab in gel, adjacent to mark near approximately 3 kb position on migration gauge. Insert a second locator tab in gel, adjacent to mark near approximately 6 kb position on migration gauge. Move migration gauge to allow insertion of locator tab in lane 4. Insert another locator tab in gel, adjacent to mark near approximately 3 kb position on migration gauge. Remove migration gauge and prepare to insert GeneStix. A GeneStix is inserted into the gel by positioning it behind the locator tab and then sliding it down the back of the tab. A GeneStix, labeled here as 4A, is placed in lane 4 to capture 5 kb and 20 kb DNA bands. A GeneStix, labeled here as 2A, is placed in lane 2 to capture DNA bands greater than 3 kb. A GeneStix, labeled here as 2B, is placed in lane 2 to block DNA bands greater than 6 kb from being captured by GeneStix 2A. Remove locator tabs by grasping the locator tab and GeneStix between the thumb and finger. Gently pull up on the locator tab without removing GeneStix. Remove all locator tabs. Return gel to the electrophoresis device. Continue electrophoresis. Desired bands have now been captured on GeneStix. Under blue light illumination, all cyber gold labeled DNA markers can be seen to have migrated past the GeneStix. Remove gel from electrophoresis device. Remove GeneStix from the gel. Insert the GeneStix into spin funnel and collection tube. Apply elution buffer to GeneStix. Place GeneStix with spin funnel and collection tube into centrifuge. Apply elution buffer to remaining GeneStix. Centrifuge to extract sample from GeneStix. Remove GeneStix with spin funnels and collection tubes. Repeat application of elution buffer to all GeneStix. Centrifuge and continue per instructions. After final centrifugation, remove and retain GeneStix. Dispose of spin funnels. Gel analysis of the captured bands, using ethidium bromide, demonstrates the utility of GeneStix. Extracted GeneStix 2A contains bands greater than 3 kb while excluding bands greater than 6 kb, which were blocked by GeneStix 2B. Extracted GeneStix 2B contains bands greater than 6 kb, which were blocked from reaching GeneStix 2A. Extracted GeneStix 4A contains the captured 5 kb and 20 kb bands. Selected bands are now ready for use in further processes. Thank you for your time. We hope that you have found our presentation about GeneStix informative. Should you need additional information, please visit our website www.princep.com.